professor in biosimilars. So I will just to make sure, is Dr. Ammar with us?
Okay. Hello, everyone. Thank you, Omar. Sorry for being late. Actually, I was in traffic going home. So please accept my apologies, everyone, and thank you for attending the session tonight. I hope you will get some information that will be useful for you during your studies and later on during your careers. I'm not sure what kind of audience do we have. Is it only students or a mix of students and pharmacy graduates? So anyways, the presentation I'm gonna deliver today is gonna be about biosimilars, okay? So I'm not sure if my screen is it?
Yes, it is. Yes, yes.
Okay. So is the audience Arab speaking, Arabic speaking, English speaking, or a mix, or what language do you prefer?
It's mainly Middle Eastern students, so it's mainly Arab, but there are some mix. So if you can just continue with English, it would be better.
Okay.
Perfect. Thank you, Doctor.
Thank you, everyone, and I'm happy to see so many people interested. I'm actually amazed. 88 people participating in this event, in this session. So my specialty is pharmaceutical biotechnology, which is a very important specialty in pharmacy, because most of the new drug entities that are actually being marketed right now are actually biological based products or biotechnology based products. A recent example of that was the COVID vaccine. The COVID vaccine is actually an mRNA vaccine developed using both biotechnology and nanotechnology. So this is one example of an application of biological agents, and the list goes on. Most of the therapeutic options or treatments currently available for cancer patients, chronic autoimmune diseases, and other diseases that were actually difficult to manage or treat are now being treated by biological agents. Now, when we talk about biological agents, we mean agents that are either protein-based or DNA or RNA-based, and mainly protein-based. And these differ than the traditional chemical agents in that their production relies on living systems. I'm not sure if you have reached that stage in your studies, but for example, when we say we want to synthesize loratadine, or we want to synthesize aspirin. We want aspirin. We want to make an aspirin tablet. Basically, what we do is that we bring chemical agents and we process them through a chemical reaction until we reach the chemical structure of aspirin. Then we formulate the drug into a tablet and package it and give it to the patients. Now, a biological protein agent, such as insulin, for example, cannot be synthesized in the same way because proteins are complex molecules and we can't synthesize them chemically. We need to rely on living cells to synthesize them for us, such as bacteria or mammalian cells. That means that their production is completely different than chemical agents. Now, biosimilars are a concept that's very important for us, especially in the MENA region, because now we have several biosimilars that are entering the market, and pharmacy students should know what biosimilars are. So I'll start by defining the different types of medications that you usually find in the pharmacy. So for example, if we take Claritin, I'm not sure if Claritin is available in all Arab countries, but Claritin is a brand name. It's a brand name for an active chemical ingredient or a drug called loratadine. Loratadine. Okay, loratadine. That drug, okay, is the original loratadine. Claritine is the original loratadine, which means that there was a company called Schering and Plough that discovered this molecule in the 80s, and they marketed this molecule in the 90s under the brand Claritine. They discovered loratadine, and they marketed in the 90s. They had exclusive protection from other companies not to imitate or copy this molecule for 20 years. So they had the market for themselves for loratadine. That type of drug is called the originator. Now, usually every drug that is discovered gets a protection, intellectual property protection, and a patent, okay, that allows the company to sell the drug by its own for a certain number of years, but doesn't allow other manufacturers to make a copy of it. Now, after this period has finished, let's say 15 years or 20 years, any manufacturer, if they know the structure of the chemical, can get the chemical and make it as a tablet and create a generic copy of the original drug. So in this case, there is a drug called loratine, which is synthesized in India, for example. And loratine is basically a generic copy, because the patent of Claritine expired in 2002, okay? After 2002, any company can just simply bring the active molecule, formulate it into a tablet, and make a copy. That's called a generic or a copy drug. So when you go into the pharmacy, you have medications that are actually separated into two types: originator drugs and copies. Now, copy drugs are a good thing because, or generic drugs, because they allow manufacture and synthesis of chemical agents at a lower cost, and this makes the medication accessible to a larger number of patients. Who can give me an example of an originator and a copy that they know about, for example? Does anyone has an idea? So Claritine is basically an originator, and loratine is a copy. They say two have the same active ingredient and the same concentration, 10 milligrams, and they are both in the same pharmaceutical form, which is a tablet. Now, I can give you another example. Or someone can give me an example. Can I see the chat here? Okay. Okay. So basically, there's lots of drugs that are generics. All manufacturers in the Arab world are generic manufacturers. All pharmaceutical manufacturers in the Arab world are generic manufacturers. That means they don't produce original drugs. They don't discover original drugs and market them. Unfortunately, we didn't reach that point. But once a drug loses its patency, they can make a copy of it. One of the famous ones in the Arab world, and it's a Jordanian company and we're proud of, is called Hikma Pharmaceuticals. Hikma Pharmaceuticals. For example, Hikma has a generic copies of most of the drugs found in the world, and they sell it all over the world. And there's lots of examples of generic drugs. Okay. So what are generics? Generics, when chemical medicines are first developed and approved, they are sold by a pharmaceutical company under a brand name, such as Claritine, or amoxicillin clavulanic acid is sold as Augmentin, okay? Advil, ibuprofen is sold under the name Advil. Once the protection of the product expires, the patent or data exclusivity, the company no longer has exclusive rights to sell the medicine by itself. Other companies can apply to manufacture and sell a copy of that medication with the same active pharmaceutical ingredient. This active pharmaceutical ingredient is the chemical agent inside the tablet that has the therapeutic potential. It's called an API. This new version is referred to as a generic. Now, is there any difference between Claritine 10 milligrams and Loratine 10 milligrams, or let's say betadine that I manufacture 10 milligram if it was a generic copy? Do you think there's a difference? Basically, once a copy applies to the regulatory agency, such as the FDA, they need to make sure that the generic copy actually has the same quality and performance as the original drug. So when you take generic drugs, be sure and safe that the drug you are taking is the same as the original one, but probably is cheaper 10 times than the originator. That's one of the benefits of the generics. So the generics has same quality and same performance. Performance, that means they have the same pharmacokinetics, they have the same therapeutic efficacy. So chemically, generic medicine is usually produced, as we said previously, through a chemical synthesis pathway. It has the same composition, qualitative and quantitative. That means the active substance is the same and the pharmaceutical form is the same. If it was a tablet, it will be the same as a tablet, syrup, inhaler, as the reference medicinal product. And it also, using pharmacokinetic data, behaves in the body in a similar manner to the originator or reference medicinal product. So chemically, there is no difference between Claritine and Loratine, and they behave in the body in the same manner. For example, their absorption, dissolution is the same. They follow the same kinetic profile, and there is no difference between them. And this allows to manufacture drugs at very lower cost, because the company who made the generic didn't spend any dollar on research and development. They just know the structure of the molecule, and they just need to formulate it into a tablet or a capsule. That's the advantage of the generic industry, is that they don't do very expensive R&D. Very expensive R&D is very good because you can innovate and create new molecules, but it also is very expensive and usually takes around $1 to $2 billion to discover one molecule and get it into the clinic. So in terms of generic names, the original medicine and the generic medicine will have different brand names, appearance, packaging, but they have the same active ingredient. So basically, they are the same medication. And this is not confusing for us as pharmacists, because we use the international non-proprietary name, which is standard for all brands of medicines. Examples of these include paracetamol. For example, in Jordan, we have Rivanin, which is very famous paracetamol generic brand, but it's the same as other brands like Adol, for example, like Panadol, the originator, and ibuprofen, and etc. So let's look at this slide. These are three different medications, brand names. We have an Amazon ibuprofen, we have major company called Major Ibuprofen, and we have Nurofen. All three are the same. They contain ibuprofen 200 milligram tablets. 200 milligram tablets. These are generics of the originator. They are copies, so you can be safe. You can use them anytime you want for the same target. Now, if we look at generic and branded drugs and compare between them, branded drugs have patent protection. So once a company discovers and spends billions on that discovery, they get a patent, or براءة اختراع, which covers them for a period of 20 years and gives them market exclusivity, so nobody can compete with them. Generic drugs are off patent. Just you can copy, once the drug has expired its patency, you can make the drug and just formulate it. The trade name here, they give it as a unique, in brand names, they give a certain brand name, but in generic drugs, they can choose whatever they want. They don't need to do animal and chemical studies in generic drugs. No clinical trials are needed. The price is cheaper than brand names. And the appearance They could be different color, for example, and shape and size, but they have the same active pharmaceutical ingredient. And usually to develop a new brand drug, you have to take around 15 years in order to have it. But if you want to develop a generic drug, you just need to copy the original drug, and it takes you around 1 to 2 years. But this is the life cycle of an originated drug. First of all, there is the discovery and screening. You start with around 10,000 entities, and then you try to see what's working, what's not. Then you have 250 entities. You start animal testing, and then you go down to 5, 10 chemical entities. You start phase 1 trials, then go to phase 2 trials, phase 3, and then you apply for regulatory approval of the drug. This life cycle is the life cycle of brand medication, originator drugs, which takes around 15 years. And that's why they are expensive, and they have to get exclusivity of 15 to 20 years. Otherwise, they won't do that process, which is very, very difficult to do. They would just stop doing business at all. But in order to make them work by this model, we give them around 15 to 20 years of patency in order to protect their drug. Now, that's all about typical chemical agents. Now, biological products are a different type of pharmaceutical medications. These biological products are protein-based, they're large, and they are produced not by chemical synthesis, they are produced by living systems such as microorganisms, yeast or bacteria, or even animal cells or mammalian cells. If we look at this slide, we can look at the structure of insulin, which is a simple protein of 5,000 Dalton. Erythropoietin is more complex. A monoclonal antibody has has a mass of 150,000 Dalton. It's a huge molecule, and this cannot be synthesized chemically. So that's why we rely on a cell. We genetically engineer that cell, and we copy it. We make it create the biological products. Examples of biological products include therapeutic proteins, such as hormones, growth factors, like human growth factor, monoclonal antibody, vaccines, and blood product. Now, if we compare a typical drug, chemical drug, with a monoclonal antibody, you can see the huge difference in size between aspirin and a monoclonal antibody. So this is a different type of science, different type of pharmaceutical manufacturing, because we're talking about large, complex proteins. Biologics are very, very successful drugs. An example of biologics is a drug called adalimumab. Adalimumab is given as a subcutaneous injection once every 2 weeks or once weekly for the treatment of rheumatoid arthritis. And its total revenue is $20 billion per year. It's called Humira. Why is it that expensive? First of all, because there is no copies of Humira. It's still protected. Actually, this year it would lose its protection. It's protected by its patency, and it has remarkable therapeutic potency against rheumatoid arthritis. Okay, so what's the problem? Can we make easily copies of biological products? That was a problem that was solved only around 10, 15 years ago. Now, the problem with biological agents is that they are proteins, and proteins are synthesized. We synthesize these proteins by using living cells, such as bacteria. So, for example, or animal cells. Now, these are synthesized inside ribosomes. And usually they are exposed to something called post-translational modification. So once a protein is synthesized, the cell usually adds different sugars on them. This sugar pattern, the way the pattern of sugars are, depends on the type of cell and the conditions of the cell. So one time it synthesizes a monoclonal antibody, it could add a certain range of sugar molecules. The next time it synthesizes it, it differs in the way it adds sugars. So it's not like chemical agents. You can't make an exact copy of the protein each time. And when you package it into a vial, it's actually a mixture of different slightly variants of the same protein. So there was a big question, how can we make copies of something that doesn't copy itself in the same way every time? So originator biologics stayed with exclusive market authority for a very long period of time until certain regulations were made to make copies of them. Let's look at this structure of a monoclonal antibody. Monoclonal antibody contains 2 heavy chains and 2 light chains. The regions, CDR regions, are the ones responsible for interacting with the antigen, and the fragment crystallizable region, or the FC region, is responsible for binding with the immune system. Usually this monoclonal antibody has also some sugar coating, and this sugar is added by the Golgi and apparatus and the endoplasmic reticulum. And not every time this sugar pattern is the same. So how can you copy the same molecule again and again? And that's why the term biosimilars have been created, in order to allow manufacturers to make copies of the same biological protein. This is related basically to the technology that we use to produce proteins. We produce proteins by using living cells. We enter the DNA of a protein into a vector or a plasmid, and then we insert it into the living cells and allow the living cells to make the protein for us. Because living cells are living organisms, they behave differently according to pH, temperature, according to their strain, according to the conditions they are living inside, and produce proteins with slightly different variations each time they do the post-translational modification in the rough endoplasmic reticulum and the Golgi apparatus. So if you go to a biological manufacturing facility, each batch and lot number will slightly be different than the other lot from the same drug, because the cell every time behaves differently and produces different glycosylation pattern, which creates variation within the same proteins. Can biological agents be copied as the way they are, in the same way as small molecule drugs? So can small molecule medicines be reproduced? Of course, we talked about this. Yes, small molecule drugs like aspirin are small in structure, can be characterized and completely defined easily, and they are entirely reproduced. However, when we go to large molecules with sugar coating and with sugar post-translational modification, we can't copy them exactly. Not like small molecule drugs. They are chemically synthesized and easily characterized. Most biological products are very complex 3-dimensional structure products that are not easily characterized, and biological products come with unique challenges that to be managed through the manufacturing process. Here the term biosimilars come. Now, when we talk about copies of small chemical agents, we say generics. When we talk about copies of original biological drugs, because we can't copy them exactly 100%, when someone creates a copy of a biological drug, it can't be 100% a copy. It can be similar, but it can't be copied. So that's why regulatory agents came up, and scientists, with the term biosimilar. That means if I have insulin, I can make a copy of that insulin, but it will never be the same as the one produced by another company. So it will be similar to that insulin, but not a copy like the generics. It's not a copy drug, it's a similar drug. So in terms of biological agents, we never have exact copies, we have biosimilar. So we use the term biosimilar instead of generics, because unlike chemical drugs, biologics can't be copied exactly. And why can't a biologic be copied exactly? Because biologics aren't made up of identical versions of the same molecule. They are mixed versions of the same, of different versions of the molecule. So biosimilars is a generic version of a biopharmaceutical that may be introduced after the patent expires of that biomolecule. However, the way this generic paradigm has been developed is different from biopharmaceuticals. For example, in the EU and the US system, the term biosimilar was coined for copies of biological agents or biopharmaceuticals. If someone wants to make the Pfizer COVID vaccine a copy of it or synthesize the same vaccine by a different manufacturing company than Pfizer, they will not call it a generic vaccine. They will call it a biosimilar, for example. These are different than small molecule drugs. And we said that there's reason for that the complexity or three-dimensional structure of biologics are responsible for this difference. Biosimilar and generic drugs are copies of original brand name medications that offer patients more affordable treatment options because they are cheaper and they are made by different competing manufacturing companies. There are, however, important differences between generic drugs and biosimilars. Generic drugs are usually chemically synthesized, while biological products, including biosimilars, are manufactured in living organisms. The active ingredients in generic drugs are small and simple. They are very easy to copy. Biologics cannot be copied exactly because they contain a mix of many variants. So that's why explains till now we don't have a biosimilar manufacturing facility in the Arab world because we didn't reach that technology yet. And biosimilars and generic drugs are both versions of the same brand name medications, and they afford more treatable options for patients. And we said they have certain differences and they differ from each other. Biosimilars differ from small molecule generic drugs by their manufacture. When we synthesize chemical agents, we usually go through chemical synthesis. However, if you want to synthesize biological drugs, you bring a human gene, you insert it into a DNA vector, you transfer that into a host cell, and then you bring a bioreactor or a biofermenter, and you grow these cells in this fermentation tank. Then after the cells create billions of copies of your protein, you break the cells and you start purifying that protein and you formulate it into a sterile, either subcutaneous or intravenous injections. Now, there is maybe in another time, we might talk about the technical requirement for approving biosimilars by regulatory body, but I hope that the main concept between differing or explaining why copies of chemical agents are different than copies of biological agents have reached everyone. And I'm ready to take any questions from the students. Thank you, everyone. And this is a slide, a short a presentation about the difference or what biosimilars actually mean. Does anyone have any questions? Can I see the chat? Please feel free to open your mic and ask بالعربي, بالإنجليزي. I'm ready to answer. No difference, but there difference in price. Panadol, okay. Panadol, brand name, and active constituent is paracetamol. Isra Muhammad, EPSF Egypt to everyone. Yes, that's right. They have a copy drug like Adol. That's true. No differences in generic drugs, but the difference is in price. Yes, that's right for active generics. Maybe different in non-active ingredients. That's true, Muhammad. In generic drugs, maybe the impurities are indifferent, but the active ingredient is the same. Yes, that's true. The excipients is different. That's also true in generic drugs. But in biosimilars, that's maybe a little bit different. Biosimilars are biological drugs that are highly similar to an existing biological therapy that is already approved and marketed by Hind, Morocco. Thank you, Hind. That's true. What are considerations need to make or develop a biosimilar? Okay, the considerations is that I'll show you one slide. When we approve a generic drug, sorry, when we approve a generic drug, we need only two main things. Okay, let's say I want to make a copy of Panadol. First of all, I need to make something called bioequivalence study. That means I bring two groups of healthy individuals. I split them into two groups. Let's say 100, 50, group 1, 50, and group 2, 50. I give them the first group, the original Panadol, okay? And the other 50, I gave them the Panda or the Adol copy. And then I take samples from their blood and measure their pharmacokinetic profile of the drug in their blood. That has to be the same behavior if it's a real copy of the originator drug. The second thing I need to do is to make sure through certain tests that they behave and they have the same quality of the originator drug. That means the solution rate is the same. They are absorbed after a certain period of time, the same manner. That's what's needed for generic drugs. But if you want to make a biosimilar, you need to do analytical studies, animal studies, small clinical studies, phase 3 type clinical studies in order to prove that you're biosimilar. So it's more expensive and more technologically advanced to prove that your drug is a copy of the biological agent if it was a biosimilar. I hope that's more clear now. How dangerous are antibiotics when used incorrectly? Okay, that's another topic for antibiotics are dangerous when used incorrectly. This is another topic, but using antibiotics incorrectly can accelerate resistance to bacteria, which means that antibiotics lose their effectiveness with years, and that means we don't have antibiotics to treat common typical infections, and we create what is known as antimicrobial resistance, which is a huge problem in the Arab world, by the way. Interchangeability of biosimilars. Now, interchangeability in the EU this year or last year or the end of last year, in the EU, you can replace, a pharmacist can replace an originator biologic with an approved biosimilar without referring to the physician. Because in the EU, they depend on the active pharmaceutical ingredient, which is the typical agent. In the US, if you want to do that, to be interchangeable, you need to do additional studies to prove that your molecule is highly similar to the original one and called interchangeable. So in the EU, you can do automatic substitution, but in the US, it's still not allowed. You mentioned that we still don't manufacture biologicals. We're lacking the technology. Yes, that's true. Till now, I think there is Julfar that they, in the UAE, they did manage to do insulin. But other than that, I'm not sure there is any other company. Why? The reason is that it needs lots of money. Of course, in the Arab world, we don't lack money. Maybe in Jordan, we lack money. But I think the Gulf countries can easily do the technology transfer by contract manufacturing. You know, first, whenever you want to transfer a technology, you cooperate with one who has that technology, you pay for the technology, you transfer it back home, and then you build your capacity. So you start knowing what this technology is. It becomes easy for you, and then you become an owner of the technology. That needs to be done in the Arab world. Like China, for example, how they took the technologies from the States and then they reverse engineered it, and now they do it even better than the original ones. So we need to invest, we need to convince governments and business people to invest in the biological industry. It's a huge industry and will keep growing. Maybe in the future, some of you will invest in that. Hikma, for example, in Jordan is investing, and maybe soon we will hear about something good. Yeah, Hikma, and Hikma also, thank you, Fadi. Hikma also operates in Algeria, so maybe they will do their first facility in Algeria, inshallah. Any other questions? Thank you. So this is a very interesting topic, and I urge you all to read more about it, to keep in track. If generics are safer than biosimilars, if a biosimilar is approved the way EU and US, because there's a paradigm. I'll show you something. This is how we approve biosimilars. We do analytical studies, animal studies, clinical immunogenicity assessment, and phase 3 clinical studies. So lots of studies to approve a biosimilar. Okay, this is the pyramid of biosimilar. If you do that, okay, you're sure that there's no problem. The whole thing is, these whole studies is just to make sure that your biosimilar is safe and doesn't create any immunogenic profile. But if your government and country doesn't follow that scheme for the approval, doesn't ask for these studies from the biosimilar and be treated like a generic, that's a big problem and could be harmful to patients, because that means it's not a biosimilar, it's a biomimic or a biocopy, fake biosimilar. How do enantiomers and diastereomers differ? This is for medicinal chemistry. I'm sorry, I can't answer that. Ya Haddad. Get a medicinal chemist and ask him about it. Okay.
Okay, so if there is no more question, first I would like to thank you, Doctor, for your short yet comprehensive lecture. I really understand and learned new information today. So we'll be sharing the attendance, and if there is no questions, I think we can end it here. Again, thank you, Doctor, for your attendance, and it was very helpful. I'm sure everyone learned something new today.
You're welcome anytime. Thank you. Thank you, everyone. And all the best.